So hormones, sorry. Yeah, okay, so hormones. How can they signal? We've already learned, just to remind you, we've already learned about, question? No. Autocrine, endocrine, and paracrine. I'm not going to spend a hell of a time on that. Remember, autocrine signaling is when you have chemicals that are released and act in the same cell. In case of hormones, it's really rare, that type of signaling, as well as for paracrine ones. Okay, it's not frequent. We can consider that type of signaling between endothelial cells, for example, in the blood vessels when they produce eicosanoids, like prostacyclins or prostaglandins, that will act on adjacent endothelial cells, but that's about it. So pretty much all hormones signal on the distant ones. They have to be released in the blood to get delivered to other cells. Now, one thing that I want to make clear, when I say released in the blood, does that mean that hormone can get from the blood into the tissue, into the extracellular fluid? Can it? Sure, yeah, why not? Absolutely. There's constant exchange between the blood and extracellular fluid, right? Okay, so we're clear on that. Now, the basic principle of regulation of hormones, we pretty much focus on this thing right here. Okay, first you gotta have to have a change, physiological change, stimulus. Think insulin. What is the change? What is the stimulus that we talk about? Glucose changes, levels of glucose rise. Sorry about that. Levels of glucose rise. Okay. What is the response? The hormone release. The pancreas. Pancreas releases insulin. What does insulin do with the glucose levels? Takes them back down. Okay, correction, right? And the parameter goes back to normal. It's negative feedback. Almost all hormones that we're going to talk about, almost all of them work through negative feedback, except for oxytocin, pretty much. Trying to think, some of the hormones in the ovarian cycle, estrogen and luteinizing hormone, at some point in females they establish positive feedback, but for a short period of time. Okay? Most of the vast majority of hormones, they have negative feedback regulation. Can you come up with something else, some other? I know that we didn't cover them, but just think about it. Any other change that you can think of? Body temperature? So, excellent. When it drops, what also decreases? Basal metabolic rate. Basal, which, so when body temperature will decrease? It's not, Yes. Like if I go to Alaska now, naked. Okay, so my body temperature will definitely decrease, right? How can we compensate it? We can increase basal metabolic rate. Hormone that does it, thyroid. Thyroid increases my basal metabolic rate. Well, if I'm in Alaska naked for a short period of time until the bear, rips me apart. But other than that, I will survive for some time. Okay? Does that make sense? So it's all, pretty much always, we talk about change, hormone, coming back. Why I pay so much attention to that, obviously, seemingly obvious matter. Because when you run into the question, if you know that insulin decreases glucose levels, you can definitely see what is the stimulus for insulin production, right? Because if it decreases them, the levels should be higher than normal, right? If parathyroid hormone increases calcium levels in the blood, then what is the stimulus? Low calcium in the blood. Does that make sense? And vice versa. If you know what the stimulus, you know what it does. Right? How do we detect hormones? I hate the first part. I love the second part. I tell you about the first part because in some labs it's still done. It's radioimmunoassay. So pretty much what you do, are you at least a little familiar with the concept of antibodies? So antibodies are proteins that can very, very specifically bind different molecules. Antibodies to, I don't know, thyroid hormone will bind only thyroid hormone and nothing else. And they will bind only human thyroid hormone, but not, say, horse thyroid hormone. Does that make sense? Good. So what we do, we have a radioactive thyroid hormone that we know concentration of. And we have sample. Okay, we mix them together and we add antibodies. Okay, so you see here, radioactive thyroid hormone binds to the radioactive thyroid, and sample thyroid hormone start to compete for antibodies. Does that make sense? Does that make sense? They start to compete for like seeds. Okay? Some radioactive ones are going to be kicked out from antibodies, and their places will be taken by the non-radioactive molecules from the sample. Does that make sense? So you have antibodies with both radioactive and non-radioactive molecules, and you have supernatant, liquid. Besides antibodies, you have those molecules too. What you do, you separate them. You separate antibodies and you separate what's not bound. And then you can see how much radioactivity you have in the antibody part and in the free part. Does that make sense? So based on that distribution, you can see what is the percentage of the hormones from your sample that bound to antibodies, and what is the percentage that's in the solution, in the free solution. And this kind of shows you the calibration curve here. So as you increase concentration of aldosterone in the test, in the sample that you're testing, you see the decrease of concentration, amount of radioactivity bound to antibodies, because incoming aldosterone will kick out the radioactive one bound to antibodies. Does that make sense on the basic level? So imagine you all are Browns fans. Okay? Just saying. And there's some Steelers fans coming through that door. And sorry about that, but they start to kick you out of your seats and you go back in the hallway. Okay? I don't have to know how many Steelers fans walked in the class. I can tell this by knowing how many of you are there. Does that make sense? That's kind of sort of an analogy of what's going on here. And if you have samples of hormone with known concentrations, you can do the curve, the calibration curve. Okay? And then you test unknown. You know where it is based on the radioactivity, and you know the concentration. Okay, so you can use standard curve to make a quantitative analysis. Sounds like a lot of pain, and it is. Although I forgot the name of that Israeli woman. When she developed, she got Nobel Prize. I think it was for insulin. She developed the first radioimmunoassay. Since then, we just go to CVS or Walgreens, buy the little glucometer, and can measure it on us without any problem. Now, ELISA, that's the immune-based assay, pretty much what you do, hormone binds to antibody. Okay? That complex of hormone and antibody is detected by another antibody. The more complexes you have, the more light special detection system is going to produce. In the very basic level. Does that make sense? Like you add more hormone, you have more complexes, and those complexes have special enzyme attached to it. It's E here. Special enzyme that will make the sample glow. The more complexes you have, the more enzyme there is, the more glowing you're going to detect. It's actually a very, very simple method in terms of how to do that. It's really simple. And it's very widely used in clinical practice. Finally, we get to the actual hormones. So those are your hypothalamic hormones, produced by that little piece of diencephalon. That is the chief controller of endocrine functions in the body. Two major types of hypothalamic hormones are releasing and inhibiting. And as you can say from the names, they either release something or inhibit something. In fact, hypothalamic hormones, most of them, signal to the anterior pituitary gland, releasing some of those hormones from anterior pituitary gland or inhibiting them. So what are those? Thyrotropin releasing hormone. Okay? Thyrotropin releasing hormone, TRH, will stimulate thyroid stimulating hormone, here thyrotropin releasing it. Corticotropin releasing hormone stimulates corticotropin. Gonadotropin releasing hormone stimulates two gonadotropins. Now going back to the words thyrotropin, corticotropin, gonadotropin, they all have something in common. What? Tropin, what does that mean? When we say something, when we say something neurotropic, what neurotropic means? Infection is neurotropic. Much simpler, neurotropic. Yeah, so it goes into the nervous system. So, thyrotropin, it goes into thyroid gland. Corticotropin goes into adrenal, or the cortex of adrenal glands. Gonadotropins signal on the gonads. Does that make sense? So that's tropin thing. Now, growth hormone releasing hormone. This is pretty straightforward. It stimulates the release of growth hormone from the pituitary gland. Right? Now, growth hormone regulation is slightly different from others. It has both releasing and inhibiting hormone. And by the balance between the releasing and inhibiting hormones, hypothalamus can regulate body growth. Because, surprise, surprise, growth hormone stimulates growth. Okay? So, this little factor here, it's called prolactin inhibiting factor, regulates the release of prolactin from anterior pituitary gland. What do you think prolactin does? Yeah, stimulates production of milk. Okay? So, we'll talk about a little bit more at the very, very end about regulation of prolactin. But essentially, when there's a loop, when baby sucks on the nipple, this hormone is inhibited, prolactin inhibiting factor. So there's no inhibition, like brakes are off, prolactin takes off and stimulates lactation. That makes sense? So it will make more sense as we go into the hypophysis, into the pituitary gland. But you do have to know these hormones and their basic function. What do they do? What do they release? What do they stimulate? Again, endocrine system is like a puzzle, because we open pieces one at a time, and unless you have the whole picture in front of you, each individual piece does not make that much sense. Okay? So since I already advertised pituitary gland for you, here it is. So you see hypothalamus, and it's directly connected by the infundibulum to the pituitary gland. It actually sits in a little depression called sella turcica in the sphenoid bone. Sella turcica, nothing fancy about this. It's direct translation is Turkish saddle. I don't know why it's Turkish saddle, just what it means. Pituitary gland has two very distinctive, very anatomically and functionally different parts. Now, when I draw pituitary gland, it usually comes out, I feel really embarrassed because when I draw it, it looks like this. So I will stick to the actual picture. Trust me, I was drawing pituitary gland, it wasn't a butt. So anterior and posterior pituitary are both connected to hypothalamus in a different way and have different function. Really, really different function. So anterior pituitary or adenic hypophysis is bigger, and it is the part that actually makes hormones. Posterior, question? Yeah, it makes hormones. Posterior pituitary or neurohypophysis, also connected to hypothalamus, but interestingly enough, it does not make any. Yes, it releases them. I compare anterior pituitary to the actual factory, actual plant. They make hormones. Posterior pituitary is more of a warehouse. It stores and releases hormones. Okay? So this is the list of hormones of anterior and posterior pituitary. So anterior makes the most. Growth hormone. Does anyone remember two hormones that regulate its production? You can go back in the slide. Two hormones that regulate growth hormone production from hypothalamus, Growth hormone, inhibiting hormone, and releasing hormone. Very good. Very, very good. Okay? Prolactin. What regulates prolactin? Prolactin in... Where'd I go? Wait. Prolactin inhibiting factor, right? Also in hypothalamus. Thyroid stimulating hormone or thyrotropin. What regulates it? Releasing hormone. Very good. TRH. Adrenocorticotropic hormone or corticotropin. CRH. These two guys, follicle stimulating and luteinizing hormones, collectively called gonadotropins. GnRH, gonadotropin releasing hormone. Does that make sense now? You see, we established the connection between the hypothalamus and anterior pituitary. What about posterior? I just told you, does it make any hormones? It stores these two hormones, antidiuretic. Now, let's look at the name, antidiuretic. What does it do? What is diuresis? Don't look there. What is diuresis? Come on, peeing. It's peeing. Diuresis is peeing. So antidiuretic hormone. What does it do? Yeah, you pee less. Make sense? ADH, not that I know of. It's kidney function, yes. It very much decreases the urine production. Okay? And it can be really, really significant effect. So this is why when you work outside for 3 hours in the heat, you come back and you take a pee, it's almost brown and it's very little of it. Right? Because this hormone keeps water inside. You see again the link. You get dehydrated, that's change. The hormone fixes that deviation by keeping water inside. Does that make sense? Oxytocin. Remember what it does? Contractions of uterus. Something else. It's kind of for kicks, huh? You can say so. If bonding is feeling good, it's produced when a parent and child are together, when men, like, it's known that oxytocin is produced during kissing. Men produce it too. During sex, during kissing. We don't know what is the exact role, probably bonding. Yes. In females, it's direct physiological, it's uterine contractions. Now, we'll get to intermediate, I'll tell you a few words about it. But if they are not produced there, they're not produced in the posterior pituitary, they just stored and released. Looking at this picture, where would you think they are produced? Where would you think these two hormones, ADH and oxytocin, are produced? And then transported to the... they're hypothalamic hormones. They made in the hypothalamus and kind of stacked in the posterior pituitary. Does that make sense? Now we don't talk about melanocyte stimulating hormone, but essentially this is the hormone that stimulates production of melanin and melanocytes in the skin, okay, and triggered by light and during just the development. Okay, that this hormone is partially responsible for us being darker or lighter hair, darker or lighter skin, and so on and so forth. Okay. Often it is related not as the hormone of intermediate zone, but the hormone of anterior pituitary. So, this gives you the outline of how things work for anterior pituitary. It looks like a lot of steps, but I will try to walk you through them in simplified, digestible manner. So if you go back for a second, you will see that all hormones of anterior pituitary are somehow controlled by hypothalamus. You know, even here. See, all hormones of anterior pituitary are somehow controlled by hypothalamus. Is that clear? Yes. Unless I specify it here. Unless I tell you, look, glucagon is the protein. Okay? I may ask you, for example, epinephrine has extracellular receptors. Which class does it belong to? Water soluble, lipid soluble. Does that make sense? Okay. Or something is like thyroid hormone is lipid soluble. Well, okay, forget it. Cholesterol is lipid soluble hormone. would it belong to peptides, amines, steroids, or proteins? Steroids, right? Because peptides, amines, and proteins are all water soluble. Things like that. But if I don't mention specifically, like if TSH is the protein or something, no. So here we go. Hypothalamus has special neurons called neuroendocrine cells. They produce hormones such as growth hormone releasing hormone or gonadotropin releasing hormone. And these hormones enter the circulation here. Okay? So this here is called primary plexus of the system of blood vessels that is called hypothalamic hypophyseal portal system. So see, that's portal system. That's this all vessels. It's the portal system. Does that make sense? All those vessels that you see on that slide, portals, they comprise portal system. So hormones enter primary plexus, then go through the veins into the secondary plexus. Okay? From secondary plexus, from the blood, they diffuse into the anterior pituitary gland and stimulate cells, of anterior pituitary. In response to that stimulation, anterior pituitary produces pituitary hormones, whatever they are, okay, and they enter the general circulation. That makes sense? So I have like this stops. Hypothalamus, neuroendocrine cells of hypothalamus, release hormones in the primary plexus. Hormones from hypothalamic hormones from primary plexus through the portal vein go into the secondary plexus. From secondary plexus, they act on the anterior pituitary gland, which then produces pituitary hormones, which go into the circulation. Does that make sense? So, some atropins, corticotropin releasing, some of the anterior pituitary, hormones of anterior pituitary gland, some of them atropins, like corticotropin, thyrotropin, gonadotropins, which means that they stimulate other hormones in those glands. Thyrotropin will stimulate thyroid gland. Corticotropin will stimulate adrenal cortex. Gonadotropins will stimulate different cells in gonads. And they will stimulate production of androgens, like testosterone in males, estrogen and progesterone in females. Okay? That makes sense? This whole idea. So we can add, actually, hypothalamus here. You see? We now have 3 axes. Same 3 axes I told you about before. Okay? Hypothalamus, pituitary, thyroid. Hypothalamus, pituitary, adrenal. Hypothalamus, pituitary, gonadal. Right? You keeping up with me? Yes. Yes. We'll get there. They made in the hypothalamus in advance, stored in the posterior pituitary. Here's the thing. I talk about anterior pituitary. Good question. When I start about anterior pituitary, I just go through the hormones that are produced there as well. We will come back to posterior and we'll chat about ADH and oxytocin. Now, one hormone that is produced by anterior pituitary gland and has a direct effect on the body is the growth hormone, or somatotropin. So as we figured out, tropin means it has effect on something. What does soma mean? Body. There you go. Effect on the body, right? It stimulates growth. That's a massive statement, right? Considering that it's called growth hormone. But in addition to growth, actually, growth stimulation is an indirect effect of growth hormone. Directly, it stimulates breakdown of fat and increase of blood glucose. So it's so-called anti-insulin hormone. Does that make sense? It increases glucose levels in the blood. That makes sense. Why lipids and fats if it's a growth hormone? Why lipids and glucose? Why breakdown of lipids? Why glucose levels in the blood go? Energy, yeah. For what? Look at the name of the hormone. For growth, yes. Now, it has an indirect effect, and we're going to get to this. So look at this scheme here. So growth hormone stimulates fat and carbohydrate metabolism, which deliver energy for growth. At the same time, it stimulates mostly liver that produces so-called insulin-like growth factors, or IGFs. These IGFs, in turn, stimulate growth. So it's not hormone per se. It stimulates insulin-like growth factors that will act on the tissues like muscle and bone, promoting growth. Does that make sense? And direct effects will provide energy, okay, for that. Does that make sense? We'll get to the graphs. What I want to focus your attention now on is negative feedback. If you will if you will take normal rat and will start to grow it, it's going to stop growing at a certain point. About 250 grams, half a pound. They're really cute like this, lab rats. The Wistar rats. If you will start injecting that rat with the growth hormone, you're going to create a mean beast that weighs more than a pound. They're really, really huge and muscular. Like really evil creatures. Well, not evil, actually, still kind. Really big creatures. Because when you inject the growth hormone, there is no negative feedback, right? You just continue supplying. So in the real life, in physiological conditions, you gotta have some negative feedback. Insulin-like growth factors provide this feedback. When they are released in the blood, so they can be considered hormones as well. When they're released in the blood, they signal on the hypothalamus and anterior pituitary. When they signal on the pituitary, they directly inhibit synthesis of growth hormone. When they signal on the hypothalamus, they decrease the gonadotropin releasing and increase gonadotropin inhibiting. So if we want to make like a short outline, that's hypothalamus, anterior pituitary, and liver. Hypothalamus produces GHRH. Pituitary gland produces growth hormone. Liver produces insulin-like growth factors. Sorry for my writing. And those same IGFs will, well, I should say, inhibit growth hormone, right? They will inhibit growth hormone releasing hormone, and they will stimulate growth hormone inhibiting hormone. Does that make sense? All of this writing in a dynamic thing. So this is oversimplified version of the flow chart to your left. the flow chart that's highlighted, all highlighted in green now, right? That makes sense? It's over. So please practice that explanation for yourself. Like doing those little, it took me what, 3 minutes? I understand you, well, it took you 3 minutes. Well, open up the slides, open up the book, do it yourself. Lay out that flow chart, okay? Now, this is going to be the last thing we're going to talk about, what stimulates and inhibits growth. Exercise and sleep are two best stimuli for growth. When you exercise, you grow. Because you give a signal, resistance training especially. You don't have enough muscles to produce enough effort to lift that barbell, you're going to grow. Of course, you have to eat also. And sleep. When your mom told you you grow when you sleep, she was right. You really grow when you sleep. Other stimuli, decreased blood glucose and fatty acids. That comes from exercising. When you exercise, you decrease those levels, and growth hormone, guess what? Provides it. So it is produced, it increases blood glucose, it increases free fatty acids, and it stimulates liver to produce growth factor, insulin-like growth factor. Protein deficiency. Now here's the problem with the protein deficiency. Growth hormone will be produced, but if it continues, you're not going to grow. Trauma. Normal response. You have to recuperate. Okay? Exercise. Testosterone and estrogen actually stimulate growth hormone. You can see it in puberty, in teenagers. When those androgens hit, growth hormone also increases and they start to grow. Sleep. Well, obviously, stimulation from the hypothalamus, ghrelin. It's a hormone of hunger. But, of course, we imply that when you're hungry, you eat. Okay. Inhibition, obviously, high blood glucose and free fatty acids. Aging. It's really hard to grow muscles and bones back when you're older. Obesity, you can link it to increased blood glucose and fatty acids. Somatostatin, or inhibitory hormone, coming from hypothalamus. IGFs, as we just saw here. Okay, IGFs inhibit it. And exogenous growth hormone. What does that mean? What mean exogenous? What does that mean, exogenous hormone? Coming from outside or inside? Exogenous. Outside. Now, coming from outside, what does that mean? Well, shoot yourself with growth hormone. So it inhibits production of your own. And when you stop shooting yourself, your growth hormone will be low. And that will have some dire consequences for your body. Does that make sense? We're going to continue talking about pathologies of the growth hormone on Monday.